Hey, quit it. Mm-mm-mm. Mm. All right, ladies and gents, let's begin. All right, because we've got a pretty chunky session today. Actually, today and Tuesday next week are pretty solid. There's an awful lot of stuff to cover, and there's a lot of details, right? So one of the challenges of this particular subject, gene expression, is kind of not getting lost in this sea of facts, right? There's a huge amount of stuff going on, right? And it's a very kind of prescriptive process, right? You need this, and this does that, and that does this, and then so on and so forth in a particular order. So just approaching it as a collection of facts is a very hard ask, right? It's kind of like learning, I don't know, Latin or like the Russian alphabet without learning Russian, right? It's just a jumble of stuff. So it's a good idea to keep in mind like the bigger picture, like of the overall process, and try and slot in each bit into that picture, right? So you don't end up just being overwhelmed by this huge morass of facts, right? Because once you get the idea of, oh, you know, you need to initiate transcription, right? You need this and this and this, and then this does that. Makes it a lot easier to remember what those things are, right? And so I'll be trying to kind of keep you within that general framework as best as I can. But at some point, you know, we're going to dive pretty deep into the weeds, right? So, you know, kind of keep that in mind. Okay, so let me just do that. Oh, that didn't help. I'm still figuring out my way around Zoom. There's, I blame Alicia, actually. She keeps showing me all these extra bells and whistles that I didn't know existed. Okay, so hopefully you can all see me just fine. So essentially, we're going to be tackling the subject that's commonly called the central dogma, right? I actually kind of dislike that term personally, because the central dogma is actually, and this, if you've had GenBio 1 with me, I'm not sure, I think there's at least a couple of you that have. You'll know that this is one of my pet favorite little rants, along with giant pandas being the most useless animal in the world. But we won't get into that because we've got too much stuff to cover. So essentially, I'm going to take pictures of these so you can study my hieroglyphics. Hypothesis, that's how you spell it. Right. This is actually the sequence hypothesis. And that is that you have DNA, mRNA. Hopefully you can see this kind of okay. Let's get this. I got my laptop perched on my lap on a cushion, so that's why it's kind of a little bit jiggly. I wanted to have one of those huge, like kind of really impressive oak-lined offices that wows and impresses students, but I didn't have time. I don't really like, you know, big heavy furniture anyway. Oops. There's an I there. Right, so basically the sequence hypothesis is that you start with DNA, right? This is your, oops, can't even spell info. What's wrong with my brain today? That's your info. mRNA is your intermediate, right? Its purpose is to serve as the basis for translation, right? So this part, I'm sure you'll know this, right? You know, I don't want to kind of patronize you because this is stuff that most likely you already know. But if you don't, this is, shit, where's my pen? Transcription. Can you see the board okay? So, Ali, can you walk Angela through the process of turning into speaker view? That should make me a lot bigger.
First of all, what do you see?
I am seeing you as the biggest person on there. It's just like the furthest part of the board I can't see.
Oh, okay. Because of reflection, or just because I need to move it around?
Yeah, like it doesn't fit in the,
okay,
it doesn't
yeah.
all fit in the frame.
I just needed to see it, but I got it.
Sorry about that. That's kind of, basically my thighs are only so long. I am quite tall, but I'll move it around so you can see it. And I'll take pictures of this and I'll upload it to Blackboard too. So you're not going to be worried about missing stuff. Right, so essentially DNA to mRNA, from information to intermediate, that process is called transcription. Right, this overall is called gene expression. Ta-da! Yeah, and I'm sorry about how clunky this is. It's you know, kind of like improv genetics, I guess. And then the final step is to make, I don't know, what we call protein. right? That's the thing that does stuff, essentially. You know, by and large, protein is not always the exception. The exception being like non-coding RNAs would probably be the best exception to that. But by and large, proteins are the things that, come on, all right, stick you there then, do stuff. And that process is translation, right? So this is really just a recap of stuff that I'm certain that you already know. The central dogma is and actually Watson and Crick, who coined the term, I can't remember which one it was, wrote a paper later on saying that he very much regretted using the word dogma because that causes all kinds of problems. Is that it only goes that away, right? So from DNA to mRNA to protein, and it can't go back. So you can't go from protein to mRNA or protein to DNA. This was really a thought experiment, right? Because they even, if you actually read the paper, I think it was 1957, 53, one of those two. I've got it somewhere. It actually says that we do not know how all this stuff works, but we kind of posit that this is how it does work, right? So it was really just a kind of a hypothesis, really, at the end of the day. And so central dogma is all about the fact that it doesn't go backwards, right? The sequence hypothesis is that it goes that away, right? And this, in general, overall, is what we call, let's see if we can do it. Yeah, gene expression, right? Actually, here's a question for you all. We covered this last lecture. What example can you think of that breaks the central dogma, which says that you can't go from protein to mRNA or RNA to DNA? Pardon me. Can anybody think of an example of something that breaks that? Yeah, retroviruses, right? So retroviruses and molecular biologists who use the enzymes from them allow this to occur, right? So retroviruses actually, or reverse transcriptase, kind of breaks the central dogma because you can go from mRNA or RNA back to DNA. Now, in the defense of Watson and Crick, retroviruses weren't even known of until, well, mid-70s, I think. HIV came about in late 70s, I think. Now, get off. And so we can hardly fault them for not knowing about it, right? But there's still no way to go from protein to either mRNA or DNA. And we'll see why when we get into the weeds of translation, why that isn't possible. Okay, so that's basically the framework that we're going to be working with. I'm trying to move this so that the reflection from my room light doesn't get in the way. This is what we're going to be working with, all this stuff. But what I'm going to show you kind of a little bit, right? We're not going to get into it in huge detail, unfortunately, because it's just too big of a subject, is that this stuff here, DNA to mRNA to protein, it's actually phenomenally more complicated than that, right? There's an awful lot of other steps between DNA and functional protein, right? Even the fact that a lot of times, and insulin is probably one of the best examples of this, the protein that is produced from expression of the insulin gene is not functional, right? It requires subsequent steps during processing and export to actually be able to do its job, right? And even when proteins are made and functional, there are things that you can do to them to stop them being functional, right? So there are a lot of proteins which can be switched on and off at will, right? So there's quite a considerable level of complexity to this process, right? And we'll touch on that a bit, but we won't really get into it in huge detail because really that's something that you would study in, say, molecular biology class, because really you could spend a lot of time just even talking about mRNA stability, right? Or protein modifications. There's protein. There it is. It's hard for me to actually see this. I've got Abigail Garcia in huge view. Oh, there we go. Ha! Now I can see what I'm doing. I'll blame that on Alicia. She didn't tell me what I was doing wrong. You know, just modifying proteins, right? I'm just joking. I'm sorry. I shouldn't pick on you. Just modifying proteins to do different things is a huge, huge subject on its own. And in fact, one of the ones that I work on, my eyebrows are looking very verdant today. That's probably why I shouldn't be able to see myself zoomed in. One of the proteins that I work on, which is a transcription factor, which we'll get to later, is modified depending on whether or not it needs to be active or not, right? And so you can actually have cellular homeostasis, right, where you kind of modify the response of the cell to changing conditions, just the same as blood sugar homeostasis or temperature homeostasis, right, which you're familiar with, sweating when you get hot, shivering when you get cold, things like that, right? So anyway, that's essentially the big picture. And so what we're going to do is we're going to go through each of those steps in turn. The first half of that today, more or less, and then we're going to finish off that transcription in eukaryotes, because eukaryotes have a lot of extra stuff going on in terms of transcription, right? Basically related to their gene structure. And we'll finish that off and then go through translation on Tuesday. Okay, so let's get up. And also I want to make sure that you have time to kind of digest all of this. So I'm going to try and manage the pace of the class. It's a little bit weird doing it online because essentially I'm working from a PowerPoint and I don't as easily get your feedback as to how quickly I'm going or not. So if you struggle at all to kind of keep up with note-taking, stuff like that, just holler and I'll slow down and give you a chance to catch up. Come on. So what we're going to do first is just give you again another kind of general view of gene expression and a little bit about gene structure, because gene structure is actually really important and it differs very much between prokaryotes and eukaryotes, and with fairly fundamental consequences in terms of transcription. Translation, not so much, but definitely in terms of transcription. And also have a natty little video to show you later, which isn't like super amazingly interesting, because its transcription is actually in some ways fairly boring. But it will help visualize things. Okay, so let's see what's useful in here, what we can use. Oh yeah, a whole bunch of RNAs here. So first off, just to put a worm in your ear in terms of molecular biology, and I've been touching on it a bunch the last few classes, is that mRNA is just one type of RNA. There are an awful lot of different RNAs, actually more RNAs than I probably know myself, right? Because I can't keep up with all the different ones that they discover. So the ones that you're probably most familiar with are these ones. Come on. Come on, let me select that. There we go. Which are RNA stands for ribosomal RNA. I'll write these up for you. And this one is super funky. This one is, actually it's called a ribozyme. See if I spelled that right. Yeah. So ribosomal RNA is actually a great example of an RNA that does something, right? An exception to the rule that proteins are the ones that do things. So ribosomal RNA is actually what catalyzes the peptide bond in translation. It's not the protein. The actual ribosome, the protein, like the structure, the protein part of it, is there as like a scaffold. It's actually the ribosomal RNA that does stuff. tRNA equals transfer RNA. And that's basically, I don't know if any, probably heard of this in terms of a language app, which is kind of ironic. But tRNA is really the Rosetta Stone, right? It allows the translation, literally, of nucleic acid, if I can spell it right. So I'm going to move this over into protein, or protein sequence, right? More than anything. Does anybody know what the real Rosetta Stone is, just out of curiosity? A little bit of history for you. Gabrielle, you can't just say yes. What is it?
Isn't it the stone that like translated, what is it? The Egyptian or some kind of, I forgot what they're called. The pictures, what are they called?
Hieroglyphics.
The hieroglyphics and
Yeah.
to other languages, so that, like it's like universal.
Yeah, I can't remember what language it went into. I don't know if it was Greek or Latin or like some, yeah, Sumerian, something like
Oh
that.
yeah,
Really, really
that's
old
right.
one. Because Sumerian is a language that we have modern like translations of, or modern, right? But hieroglyphic we didn't until we found the Rosetta Stone. So yeah, if you ever want like a name for something to translate from something which looks like gibberish into something you know, like we always use the phrase a Rosetta Stone. So that's why, because until then there's no way that people could figure out what hieroglyphics meant. Yes, essentially it's a decoder. Anyway, I always just think that's cool. I'm a big fan of history personally. snRNAs are small nuclear RNAs, which we'll get to later. Those are involved in splicing. Don't know what snow RNAs are. miRNAs are microRNAs. There's Pee-wee RNAs. There's small interfering RNAs. There's just a whole panoply of RNAs, right? So there's a whole lot of stuff going on in the RNA world, essentially. Bumpty bum. We don't need to talk about this. So this is really just like summaries type stuff in terms of like some of the things that are involved in gene expression, some of the words involved, right? Polypeptides are really like chains of amino acids, right? Which eventually are folded and form proteins. And those amino acids look like that, right? So this is really just good basic information if you haven't done GenBio 1 recently, or some of that information is just, you know, fallen out of your head temporarily. Good recap type stuff. And again, this is essentially the basics of what we'll be getting into next Tuesday in terms of translation. We'll come back to this scary table, and really, I don't want you to worry about memorizing this because I view that as being cruel and unusual punishment. But we will come back to this when we talk about protein properties, right? And the effect of different side chains on protein properties. Because essentially that thing there, that R, not the pirate's favorite word. Yeah, I know. So, I mean, it's always a bit of a challenge because there's some students that have gone straight through the sequence. So it's probably a couple of semesters ago at most that they did GymBio1, and others that might not have done it for like 10 years or so. So if something, you know, you had to memorize it with Dr. Wise, oh, I'm sorry. Should have taken it with me then, shouldn't you? If I was here at that time. Oh, okay, fair enough. Yeah, so essentially this is essentially the backbone, right, of the polypeptide chain. It's just a sequence of amines, carbons, you know, what were carboxyl groups. And that's where the whole amino acid name comes from, essentially. It's the amino group at the beginning and the acid group at the end. And yeah, when you make that polypeptide chain, right, essentially the only thing that differentiates one polypeptide chain from another is what R groups are present and in what order are those present in, right? And that can make huge differences in terms of structure and function, right? Between, you know, proteins like hemoglobin, which are designed, well, not designed, have evolved to do a very certain job, which is to carry oxygen around, to things like myosin, right, which allows me to do this, right, which is a very linear kind of twisted chain type protein. And so basically these R groups and their properties are what determine the structure and function of that protein. That's probably the best way of putting it. And you can see why, because A, they vary hugely in size, right, from glycine, which is just a hydrogen atom, to arginine, which is this long chain, charged chain of carbons and amine groups, right? So depending on which ones you have and what order you have them, your protein structure is going to change and its function is going to be potentially very different. Okay, so I'm going to bounce around a little bit here. So essentially there's, just kind of refresh my memory about what's where. There's a couple of things going on in terms of gene structure and gene expression. All right, we've talked a lot about the directionality of DNA and mRNA, right? The whole 5' to 3' deal. Well, that also follows through into protein directionality. As if you go back up here, right, you can only add amino acids to the C-terminal of a growing chain, or even just any amino acid. So just as in DNA and RNA, right, we have a 5' end and a 3' end, and stuff can only get added to the 3' end of either of those, right? We also have two different ends in protein. We have what's called an N-terminal end, which is where the amine group is. If you come up here, right? So the very first amino acid, which is typically methionine, in fact, it always is methionine unless it's cut off later on. But anyway, the very first amino acid, its amine group is the N-terminus, right? Or the N-terminal or N-end of the protein. And it only gets made in the C-terminal direction. And the C is from carboxyl, right? So if you come back down here, and essentially you're going to be, you make DNA in a very specific direction when you undergo DNA replication, right? It's always add nucleotides to the 3' end. When you transcribe that into mRNA, you're going to use the template strand, right? This is actually a little bit of a tricky concept, which we'll get to in a little bit. You transcribe the template strand also in a 5' to 3' direction, right? You add new RNA nucleotides on at the 3' end. And when you translate that mRNA into protein, you start at the 5' end, give or take, right? Not right at the end, but towards the 5' end. and you start making your polypeptide chain in an N to C terminal direction, right? So even though the final product in terms of protein could look like a squiggle jumbled mess, right? And these are typically large, very complex structures, right? They're three-dimensional structures, right? Whether they're long and thin, like tubulin. Actually, tubulin is a little ball, but it's got lots of little balls stuck together to make a chain, right? Or actomyosin, which basically kind of slides over each other like this. Really does. Like that. Or even better, these are actually some of my favorite proteins, and it sounds super, super nerdy to say you have a favorite protein, but I'm going to write it here just so it's in. There you go. These are what are called cargo transporting proteins. That's how you get, so neurons, right, at my fingertip. You know where the cell body for that neuron exists? About here in my spine, give or take, right? And so to get stuff down to my fingertips or even down to my toes, right, which is probably a longer journey, there are actually proteins which walk like that along the cytoskeleton of your axons or dendrites to get down all that far. They're super cool. Anyway, that's a bit of a kind of a tangent, but protein structure is very complex, three-dimensional, and it results from a linear, kind of two-dimensional, I guess, barely, right? Chain of polypeptides. It's like going from a long Mardi Gras chain, right, or necklace, right, that's strung out in like a long kind of line, to a jumbled mess of Mardi Gras necklace that's stuffed in your sock drawer or something. Hang on, I want to check, see if that's is that my favorite one? Oh, that's kind of cool. Yeah, I mean, that's neat. Let me show you an even cooler one though. Sorry, this is, it's okay, but this one's actually just quite fun. Is it, trying to remember if they're in Holland or Denmark. I think they're in Holland. Yeah, there you go. Watch that later. It's kind of fun. Anyway, getting back on track. And essentially you go from that very linear two-dimensional primary structure through a series of secondary structures, which are these alpha helices and beta pleated sheets, basically, are the main structures, as well as a whole bunch of unstructured kind of loops and connecting bits and pieces. Those together, those secondary structures, make your tertiary structure, which is typically, but not always, like the final version of that protein. You also have an extra level of structure where you stick lots of different proteins together to get function, such as hemoglobin. I guess maybe even histones kind of count as that as well, because you need multiples of them to do the job that histones do. Anyway, so even though this looks like a incredibly complex structure, if you wish to do so, and it's kind of, I guess you could make this into a game, right? You could actually follow the chain, like the original linear chain from the amine group, the N-terminal here, all the way through this, like that. I don't know, I'm not going to do it because it would just take too long. all the way to the very end, which is the C terminal, the carboxyl group at the end, right? So just kind of hammer home that point. However, we have 2 strands with DNA. So potentially both strands can be coding strands, right? And let me move this over. So you can have a gene which goes this way and a gene which goes that way. You can have them like so. You can actually have them overlap. That's kind of rarer because, I don't know, you have to have the right amino acid sequence kind of shared but opposite, if you see what I mean, right? But you do have genes going in both directions, right? So you use both strands as coding strands, right? And I'm just, do we have a good view of that? We don't, right? So I just want to go over that in a little bit, so that, because this is a little bit of a tricky concept, I guess. Let's stick that as far away as I can get it without it falling off. Okay, so we have our DNA anti-parallel double helix, right? And I always draw it 5' to 3' like on the top. It doesn't really matter which way around you do it. So our genes always go kind of like in that direction. They always go 5' to 3' because of that directionality deal, right? Now, question to the group. Can you use this strand as the template to make your mRNA? Think about what direction mRNA must be made in. Right, this strand is going from 5' to 3', right? And so the gene is going from 5' to 3'. What direction do you have to make your mRNA in? Five prime, 3'. Good. Now, can you, and you know that DNA and mRNA is an anti-parallel deal, right? So would you be able to use this as your template to be able to make that mRNA and still go in that direction? No. So what you do instead is you make your mRNA, and this is your using the other strand, the complementary strand, as your template. So this is often called coding strand, because that's what encodes the information. And this is your template strand. Oops, there's an R in there, promise. And that's because, as Angela said, your mRNA also has to be made in a 5' to 3' direction, right? And so the only way that you're going to get a copy of this is if you use the opposite strand, the template strand, as, well, the complementary strand as your template. Now, that also applies to genes go in the other direction. right? So this is very much a context-dependent term. Which one is a coding strand and template depends on which gene we're talking about here, right? Because this is also the coding strand, but for this gene, right? For exactly the same reason, right? If it helps, you just turn it around, right? Looks exactly the same. That's actually a plus of using a little whiteboard rather than a big one. Can't take them off the wall. And so because this gene is going in that direction, the mRNA needs to be synthesized in that direction, and it uses the opposite strand as the template, right? So depending on which, can you see a little bit better now? I'm too small. Unfortunately, I can't make myself any bigger. Let's see, let's just try something. How about that? Better? Abigail, I can't hear you for some reason. Don't know if you're asking a question. Okay. So obviously, like I said before, you can just turn it around and it'll look exactly the same, right? Other than the writing being upside down. Oh, okay. Sorry, I just get a notification that someone's talking at the top. And so I didn't know if it was just that I wasn't hearing you. And because we have these two strands, obviously you can have this being the coding strand here, but also you can have the opposite strand also being the coding strand. It just varies which one then acts as the template, depending on whether you're going that way or if you're going that way. Does that make sense? Any questions? No? Cool. I'll take a picture of that. Yeah. My trusty rusty camera. Oh no, I have enough of those, thanks, Angela. And I've got past the whole actually, no, my kids still do drive me crazy. I don't think that's an age-dependent deal. I think it's just having children. Let's get this back up again. You know what you need to do, Angela? That will calm them right down. Give them a load of sugar. No, actually, don't do that. That would be a terrible idea. Okay. Yeah, I know. Kids are awesome, I guess. Well, they are, but sometimes they're less awesome than others. All right, so that's kind of like the kind of big picture overview. So essentially, we're kind of going from you know, like the nuts and bolts, looking at a little bit bigger picture in terms of structure and organization. And now there's a little bit of comparing and contrasting essentially between prokaryotes and eukaryotes, right? So come over here. Here we go. Prokaryotes are relatively simple, relatively, right? We want to be careful with that because they do some crazy stuff. Ooh. Coffee's running out. And so for prokaryotes, the protein is collinear with the coding region, right? This may make sense, right? It seems quite obvious. You're going to have, for every you know, bit of information in the DNA, you'll have a matching bit of information or matching amino acid in the protein, right? That's pretty easy to get conceptually wise, right? Actually, let's get this rubbed off. So I can use that in a little bit. However, eukaryotes are, come on, very much not the case. And so we have the same deal in terms of direction of transcription as we do with prokaryotes, right? That principle does not change. Pardon me. But in eukaryotes, their gene structure is much, much more complex, right? So, and please do stop me and ask questions if stuff doesn't make sense. So essentially in eukaryotes, right, we have a bunch of stuff going on, right? This is always so essentially a eukaryotic gene, actually, I mean prokaryotic too, consists of two different things. Let's write this down. I'll move this over so I can see what I'm typing. So essentially each gene, whatever it may be, right? And this actually kind of even applies for non-coding genes as well. It's just the non, anyway, don't worry about those. Essentially there's two regions. One is the regulatory region. That's what's called the promoter over here. And this is the bit that is involved in essentially the initiation of transcription, right? Whether or not that gene is turned off, the bit that RNA polymerase and all the other jazz binds to, stuff like that, which controls the expression of the gene. There are other bits which also sort of control the expression of the gene overall, but we'll get into those later. And then you have the kind of the coding region, right, which is the bit that is transcribed. So it's not necessarily just the bit which codes for protein, but it's the bit that is transcribed into mRNA, right? It's probably a better way of putting it. So in eukaryotes, you have this promoter, and that can go for a long way, potentially millions of bases kind of upstream, right? Because again, we've got direction. So if you think about, you know, a gene as a river, right? The headwaters are going to be the regulatory region, and then it's going to flow through, and the end of it is going to be downstream somewhere, right? So we use those terms a lot in terms of genetics, right? Upstream and downstream. So the regulatory region is upstream, it's plus in terms of numbers, and then we have the transcribed region, which is downstream of that. So a little bit of terminology. And so that's true here, right? So there will be a regulatory region kind of to the left or upstream of this TRPA gene. But in prokaryotes, the amino acid sequence maps directly to the DNA sequence. In eukaryotes, that's not the case because we have things called introns, right? So let's put a little bit more terminology up here. So promoter, oops, isn't someone who, you know, amps up a band and gets lots of ticket sales. Promoter is basically where the transcription machinery, both in terms of initiation and actual binding of RNA polymerase, occurs. Right? UTR equals untranscribed. Oh no, sorry, untranslated. And that's part of the mRNA, but not part of the protein. Oh, not translated. Probably a better way of putting it. Exon is protein coding sequence, right? So essentially, it's a, you know, a series of codons or 3-letter words which then get translated into amino acids. And again, remember, this is initially at least all part of the transcribed RNA, right? We'll get to what happens to those other bits Tuesday. And introns are essentially spaces. So you can have in eukaryotes, you can have genes which are crazy huge, right? For example, 100,000 bases, you know, 100 kilobases in terms of size, like monster genes. And of that, maybe 10, 15, 20% is actually coding for protein, right? Because the bulk of eukaryotic genes is actually made up of introns, right? And we'll get to why those introns exist and their purpose when we talk about splicing on Tuesday. So if you want to get a heads up on that, have a look at that PowerPoint before we meet on Tuesday. So essentially, the protein only corresponds to this bit, plus this bit, plus this bit, plus this bit, right? Exon 1, 2, 3, and 4 are what code for protein. All the rest of this, the UTRs and the introns and the poly-A signal and so on, do not code for protein, right? They're involved in control of translation, RNA stability, alternative forms of that protein, right, which we'll talk about when we get to splicing. There's a lot more bells and whistles with eukaryotes than prokaryotes. And that's actually a bit of a general theme, right? And one could, in many ways, kind of extrapolate that to complexity, right? So eukaryotes in general, not all of them, but in general are more complex than bacteria, right? And part of the reason for that is that we have many more ways to control gene expression. And so if you think back to the whole sequence hypothesis gene expression deal, right? DNA, mRNA, protein. Prokaryotes have a fairly simple array of processes in here in terms of transcription and translation. Eukaryotes have many, 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 many more ways of controlling gene expression overall, right? We're talking about the getting the end functional protein product, right? There are so many more steps along that path in eukaryotes than there are in prokaryotes, right? So there's many more ways in which you can tweak it and control it to get more complex outcomes than you can in prokaryotes, right? So think of this kind of more complex gene structure as part of that larger more complex organismal structure, right? Is kind of one way of thinking about it. Oh, even less, 5%. Whew. Okay, so essentially a little bit more recap. Again, you may feel that we're kind of covering stuff that we've already covered, and that is actually the case. Part of that is because some of these things are very central tenets, right, that we need to reinforce, right? So essentially, oh, I'm sorry. Perhaps I haven't been sleeping well or something. I don't know. I'm absolutely wiped out. Some differences between RNA and DNA. You already know this, right? It's ribonucleic acid, and it's called ribonucleic acid because it doesn't have a hydroxyl group removed. That's essentially what deoxy means in DNA, right? Deoxy means that that OH has been removed and replaced with an H. So in RNA, which is just ribonucleic acid, that OH is present, right? So there's an OH attached to both second and third prime carbons. Now, that second prime OH doesn't actually do anything. Well, ooh, yeah, I probably ought to be careful about what I say about that. Don't think it makes any difference in terms of, no, actually it can do. Yeah, it can actually be used in terms of secondary structures in RNA. Generally not. It is used in splicing, which we'll see a little later on. But it also makes RNA much more vulnerable to degradation. One of the reasons, it being single-stranded is the other one, right? So RNA is generally speaking very fragile compared with DNA. DNA has its own, you know, quirks, but RNA is like you look at it funny and poof, it's gone. Actually, real life example, I'm trying to get some gene expression work done, and I made some RNA a couple of years ago with a student to do a large scale transcriptomics experiment. The stuff that we didn't use because, you know, the sequence in place didn't need it. We stick it in the -80 freezer, so that's -80 centigrade, and it's really cold in there. It's kind of one of those places that you don't want to touch things with your bare hands because they stick. And anyway, so we want to do some more work. Dug those samples out of the -80, again, -80 centigrade. So this is really, really, really cold. And sent them to the sequence in place, and they said, "Sorry, mate, they're junk." So that RNA had actually degraded in just 2 years at -80 degrees centigrade to the point where they can't use it to do reverse transcription, actually, to make cDNA. So I've got to make the samples all over again, which is a real pain in the proverbial. Actually going into work tomorrow to G my worms along a little bit so that they're nice and happy. So RNA is very unstable. It is a very fragile molecule. And actually, in many ways, that's a benefit, right? Because if you want to turn off gene expression, you don't want the intermediate, what you're going to be using to make protein, to hang around for any longer than you want it to. So you actually do want mRNA in some respects to be intentionally short-lived. So that's another way of thinking about it. And obviously the other difference is we have a slightly different base where thymine is no longer thymine, it's uracil, right? It's single-stranded, but it doesn't have to say a single-stranded molecule, right? It can, have we got a good example? I thought I had a tRNA in here somewhere. Let's have a look, see if I can find one online. Now let's go back. That was a nice picture. All right, let's see if I can get out of this. So look at that. Absolutely crazy, right? If you want to look at it in, let's make it bigger. Can we make it bigger? Or is this going to drop me in somewhere I don't want to go? Yeah, that's probably going to drop me somewhere I don't want to go. Let's not do that. No, I like the other one better. So RNA, even though it is single-stranded, typically, can also form lots of two-dimensional structures. So it can base pair with itself. It can produce these stems, right? Where this is actually, if you look at it closely, that's double-stranded RNA, and loops, right? And so an actual tRNA, this is like a kind of 2D model essentially, looks like a clover leaf, right? But when you look at it in 3 dimensions, you can see it actually has a far more complex structure, right? And that's not the only RNA that forms weird and wacky structures. Lots of them do. And in fact, a lot of that is involved in stability or not, right? Controlling how stable tRNAs or RNAs in general are. Okay, let's get back to where I was at. Okay, so again, this is kind of somewhat of a recap, right? RNA polymerase is obviously the thing that does things in terms of transcription. It's not the only enzyme involved, or the only protein, sorry. There are lots of other proteins involved in transcription, not just the control of transcription, but also the process of it too. And there are different RNA polymerases, right? So RNA polymerase 2 is the one which does mRNA and also small nuclear RNAs, which we'll come to Tuesday. And other RNA polymerases do other aspects or other RNAs, right? So ribosomal RNA is RNA polymerase 1, tRNAs and some rRNAs are done by RNA polymerase 3. So again, getting back to this idea of multiple proteins working together, the actual thing that transcribes RNA is actually called a holoenzyme, right? And holoenzymes are complexes of multiple proteins stuck together to make like one monster protein. It's kind of like oh no, I'm showing my age if I talk about like transformers sticking together to make, you know, bigger transformers. But that's kind of the idea, essentially. Damn it, I wanted that. Alt H. Let's see if that works. No, it doesn't. These things aren't as... Okay, I can't get back to the chat. If anyone's chatting at me, you might need to shout at me to get my attention because I can't get the chat back up for some reason. Yeah, so there's actually a whole bunch of proteins that come together to form an enzyme to actually produce this transcript. And so the process of doing so is similar to DNA replication. The difference being is that the mRNA doesn't stay attached, right? So it does form using base pair complementarity, right? So you have C's base pairing with G's, U's, which equivalent of thymine, base pairing with A's and so on, right? And so that allows that specificity of transcription, right? Because you don't want to make a transcript which doesn't reflect the information in the gene itself, right? And essentially these bonds here between the sugar and the phosphate group, again, those are called phosphodiester bonds. Okay, so to finish up, at least for this little section, right, what I want to do is just talk a little bit about structure, initiation, and show you a little video, right? And then we'll have a break into about 12:10, 12:15 or so. So essentially, and this is a neat picture, and we'll actually show you, you'll see this in the video. Well, RNA polymerase 2, like the holoenzyme, right? Because again, this is like a whole bunch of proteins stuck together. What it does is it unwinds, so it takes the DNA, how we can do it, like that's in the helix, and it unwinds it so that it's single-stranded, right? And then using that template strand as it's scooching along, it makes the mRNA transcript by allowing RNA nucleotides to come in, base pair with that free base on the DNA template strand, and then it just goes zip, and it makes the phosphodiester bond between them. And so it runs along, basically unzipping DNA as it goes, and it allows it to recoil behind it at the same time. So it avoids the whole issue of supercoiling. Let's see if I can find that. Oh, Voltron. Yeah, that's the one. Sorry, you know, guess my age is affecting me. Can't remember these so well. Let's see. No, really. That's what I want to share. There we go. Okay, so going back to WeHi. Actually, no, don't want to do that. I want to do this. And let's slow it down a touch. Stats for nerds. Oh, that's nice. Okay. That's weird. Trying to figure out why I can't. Ah, come on. I used to be able to slow things down a little bit. Why can I not do that? Oh, there we go.
Because this is an old video.
No, I was just clicking on
Oh,
the
there
wrong
it
thing.
is.
Dang nabbit. All right, that'll do. So you don't have to have the sound going if you don't want to. But essentially what we're looking at here is, you know, obviously a bit of DNA, stuff floating around, blah, blah, blah. But these proteins here are proteins which are involved in the recruitment of the RNA polymerase holoenzyme, right? So RNA polymerase itself is, as I like to put it, dumb as a box of bricks, right? It doesn't know where to start transcription, or can I get chat back up? I still can't get chat. Or, you know, what gene needs expressing or whatever, right? Its job is simply to make mRNA. And so it relies on other proteins called transcription factors to tell it where to go, and when it gets there, to hold it there, right? So the actual elongation part of transcription is really boring, right? There's really not very much going on other than just making mRNA. The initiation and termination to some degree, but certainly the initiation of transcription, that's where all of the complexity is. In fact, in many ways, a large chunk of the complexity of gene expression, right, turning information in DNA into functional protein, a lot of that complexity is in the control of the initiation of transcription. There are other bells and whistles and tweaks and knobs elsewhere, but the biggest one I would say would be what genes get expressed, right? And so these proteins bind to specific regions in the promoter and, you know, the regulatory region in general, and they recruit RNA polymerase to come in and give it a kick up the butt to get started. So that's essentially what's happening here, right? These transcription factors are bound, recognized in the sequence, and they cause this complex to form. No. The music sounds really weird when it's slowed down. Right, so this is that complex of proteins, and there, I don't know how many there are at least in there, at least 10 or 11 different proteins which are involved in just the initiation of transcription in any given gene, right? So this is the, which one's the yellow one is the RNA polymerase, I think, in this, I can't remember. Where's the color? Oh no, it's the cyan or turquoise one. It's in there somewhere. So this binding is the actual like first step, right? Well, the first step is transcription factors binding to tell it where to go. Then the second step is the RNA polymerase comes in and it binds, and we get this large complex of proteins formed, which are involved in kind of retaining it there. And then the third step is activation of RNA polymerase, right? And so at present, that hasn't happened. And so if you don't do it, again, you don't get gene expression or transcription from that gene. And so the final step is the activation. And that typically occurs with a protein bound somewhere else, right? And that's what you see happening there. And so let's go back a little bit. There are regions like activator regions where proteins bind that will activate RNA polymerase. Sometimes they're a little bit closer. Sometimes they can be very, very, very far away, right? And those will cause the DNA to bend round, right, to loop around. And then that protein, which is bound here, will touch the RNA polymerase, and somehow, I don't actually know how, change its conformation into the active form, and then it will start its process, right? And that's what you're seeing here. You can see it bend round. That's the protein bound there. Touches. This is mediated protein. Oh, crap, it's gone already. Yeah, it's really fast, even at half speed. Come on, back you go. And so in that complex, there's our RNA polymerase holoenzyme, and off it goes. And that's pretty much it. This is not a super interesting. Trundle, trundle, trundle. I just want to find the blow up. Here we go. This is actually kind of neat. And so here you can see that localized kind of unwinding of the DNA, right? So that, and if this gene is going in that direction from right to left on the screen, then the template strand will be the one at the bottom, right? And so, I think that's right. Yeah, it should be. And so essentially those nucleotides are coming in and they're forming this growing chain, pardon me, coming out the end of the RNA polymerase. It doesn't stay bound, not for very long at least. It's only during the initial synthesis part that is the case. And so if we look at that in, does it go in more detail? Well, that's pretty much it. Like I said, the elongation part is not super interesting. And then it just ends up over here somewhere. And at some point, it's got to stop, because if it doesn't, you're going to transcribe your entire chromosome, and that's an enormous waste of resources. And also, you probably don't want to transcribe every gene on your chromosome at once, right? And so at some point, there's a signal, and that signal varies between prokaryotes and eukaryotes, tells the RNA polymerase to stop, right? And then it dissociates from the DNA. The mRNA transcript is then kind of ejected and gets booted out of the nucleus to the cytoplasm. In eukaryotes, prokaryotes, it's different. Does anybody have any questions so far? Let's see if I can. I don't know why chat is not. Oh, there it is. It's coming up. Questions? I mean, the basics of it are fairly straightforward. It's when you start getting into the details that things get a little trippy. And so essentially there are in both prokaryote and eukaryote genes, what's called a core promoter, right? And so a core promoter is the bit of the DNA upstream of the coding region where RNA polymerase binds, right? And so in bacteria or prokaryotes, right, you've got your transcription start down here, and you have your upstream regulatory sequences. You have what's called a TARDA box, and there's a TTGACA box. These are called consensus sequences. So essentially they're consensus sequences because if you take a whole bunch of immediately upstream regions from that species and you line them up next to each other, most of them will have a T in that first position. Most of them will have a T in that position, right? And so these sequences, they're not exact, right? So you can look in the first one, it's TATGTT, right? So there's a couple of differences. Here it's CATGAT. Again, what's that? A couple of differences, right? So they're not the exact same sequence, but overall, they share mostly the same nucleotides, right? Bases in them, right? And in prokaryotes, these are a very specific distance, right? And this distance is important because that's kind of the size of the RNA polymerase which is coming to bind and the transcription factors involved, right? So that spacing is really important. And you'll also notice that both the -10 and the -35 sequences are AT rich, right? So I think the -10, I can't remember 100% sure. The -10 is actually where that initial uncoiling occurs in transcription. I think it is, not 100% sure. But it would make sense if they were. And this is just a neat kind of visualization of what a consensus sequence looks like. So essentially, if you look in a position, how the size of the letter equals how often you find it there, right? So in these positions, for example, you always find a C and a C, right? Other ones, you may find other ones, but one letter in particular is more predominant, right? And so that's essentially what we mean by consensus sequence. And I think that's probably a good point to stop right here because we're going to start getting into bigger details, right? So I'm going to pause the recording and let's get back in 15 minutes. Let's say 12:30. Does that sound good? Awesome. How do I pause the recording? That's the next question. All right, see you in 15. Good stuff. Get the recording going again. All right, hope you had a chance to stretch your legs, get some refreshments. We need to go back to this. Okay, so let's see if we can get chat up. Can this time. All right, so essentially, in terms of promoters, right? So again, promoters are this region here upstream of the coding region or the transcribed region, which are involved in RNA polymerase binding and initiation. So obviously, depending on how well those, where are we at? Here we go. How well those bind, how well transcription factors bind, how well RNA polymerase binds, and so on, determines a lot of the outcome of gene expression from that gene, right? So you can have what are called strong and weak promoters, right, which arise through mutation, and they're essentially variations, at least in prokaryotes, variations of these consensus sequences, right? So the closer you or a promoter hews to these consensus sequences, typically the stronger it is. And by stronger, we mean more product being produced from that gene in terms of mRNA. And obviously there are differences in how much mRNA one would want from a gene. So if you, for example, if a bacterium comes into a new food source, right, you want to utilize that really quickly, right? So you want to put out a lot of proteins that can metabolize that food source, right? On the flip side, there are some things which, you know, you need all the time, but you don't need a lot of them, such as, say, ribosomes, right? So you might have a lower but more consistent gene expression from those particular genes, right? It just depends on the context, right? But on the flip side, if you're not on that particular food source as a bacterium, you don't want to produce those proteins because they don't have anything to work on, right? So you need to be able to turn those things off as well. Okay, so essentially, and this is where we start getting into the details, right? And this gets even more so for eukaryotes. So it's again a little simpler for prokaryotes, and they have their own quirks too. And so essentially, the initiation part in bacteria is driven by sigma factors, right? So these are essentially, it's a transcription factor which allows RNA polymerase to bind to that promoter, right? That holoenzyme, you know, that complex of different proteins which forms the RNA polymerase, then, as I said, unwinds that locally, that promoter region, right? Presumably around one of those consensus sequences, and then, you know, does its job, basically, right? Synthesizing the new RNA transcript. So relatively straightforward in bacteria. We'll get into some of the control of gene expression in bacteria and eukaryotes too after exam 3 next week. So that will be the topic for, when's that, next Thursday? Let's double check. Yeah, so we'll start on that next Thursday and continue that. Oh crap, there's not actually a whole lot left of time in the semester. Wow. So I'm just going to show, always just remind myself exactly what it is that we're going to be studying. Come on. There we go. So essentially this is what we're doing right now. It's the 9th. Finishing up transcription and then translation on Tuesday. And then Thursday, we start the last section, which is, well, we have the exam as well, which is control of gene expression in eukaryotes to begin with. And we've got a couple of different examples there, and prokaryotes, and then some kind of, you know, bigger picture stuff to finish up with. Right, so there's not a whole lot left time-wise in the semester, to be honest. It's gone by really, really fast. Okay, so stop that one. Oh, well, that just pumps me out of that. Great. Okay, in eukaryotes, however, let's get chat back up. Come on. This is actually, anyway, if you got something in the chat, just give me a squeak and I'll go look at it. I can't see it right now for some reason while I'm sharing. In eukaryotes, we've got a similar kind of idea. We've got a consensus region that's also called a Tata box, right? Because it, again, largely consists of A's and T's. But one of the big differences in eukaryotes is that it's a lot more complex, right? So the distance is more varied, right? In terms of the spacing from the start of transcription to that RNA polymerase kind of recognition box, right? The Tata box. And the promoter sequences in general in eukaryotes are much more complex and larger than they are in prokaryotes. And that essentially just ties into that complexity deal that we were talking about earlier. You know,
I'm sorry to interrupt, but did you start the recorder?
Let me just double check. I think I did. Yes, I did. Thank you though for the reminder. Yeah, actually one cool thing about Zoom is that it automatically starts recording at the beginning of any session. So at least I don't have to remember that. That's a plus. And also there are a bunch of general transcription factors in eukaryotes, right? So these aren't specific transcription factors that are specific to particular genes, right? Those we'll talk about more when we talk about control of gene expression in eukaryotes later. These are more the equivalent of the sigma factor in prokaryotes, right? So they're actually, let's see if I can get out of this. Let's see. No, that doesn't work. I'm just trying to. Hey, Gabrielle, no worries. Just catch up as you can later on. This will be up online by this afternoon. I'm just having issues with my chat box disappearing and not being able to get it. This time it worked. Who knows? It's kind of a little bit flaky. All right. Anyway, so there are a whole gamut of transcription factors, huge numbers of them, which are involved in turning on specific genes, right? But these ones are more generic, right? They are just involved in getting RNA polymerase stabilized and bound, right? And so essentially in eukaryotes, this is a very kind of simplified version, right? And again, now we can differentiate between what's called a core promoter, which is really about RNA polymerase binding and initiation and that kind of jazz. That's where the Tata box is. And the regulatory promoter, right, which is more involved in kind of essentially fine-tuning that, controlling activation, controlling a lot of repression activation, cell-specific or tissue-specific activity of genes. That's in the regulatory promoter, right? So again, you've got that much smaller core promoter, right? And that can be as little as 200 base pairs, 300 base pairs or so. And you have a much, much larger, more varied regulatory promoter, which is involved more in the specifics of gene expression of that particular gene, right? And so in that core promoter, you've got your Tata box. And unsurprisingly, you have a Tata box binding protein. That's what TBP stands for. And so essentially, Tata box binding protein there, and one of the transcription factors, TF2D, those form a complex. They bind to the Tata box. That's what they recognize. And then essentially all the other stuff that, if you remember that video that I showed, there's a whole bunch of proteins that kind of assemble and stick and things like that. All of those assemble with RNA polymerase II, the holoenzyme, to create this complex, right? And one of the key proteins in this complex is the mediator protein, which is this one up here. It's kind of in a light purple color. mauve, maybe. And this protein essentially links the complex bound to the core promoter to any proteins that are present in the regulatory promoter, right? Both in terms of inhibition and activation, right? And so that protein, the mediator protein, is what that enhancer or activator kind of bends round and touches, right? Mediator kind of mediates essentially, right? The interaction between the things that turn on transcription and the actual machinery of transcription itself, right? How does that work for you all? Does that make sense? So you can see it's a little bit more complicated than in bacteria. Jasmine. Sorry, I've just seen my psycho princess. She doesn't come out very much. Oh, she's around hunting her sister for some reason. What do you want? Come. Come up here. No, she doesn't care. Anyway, let's see if you can see her. There she is. Jasmine, say hi. She probably wants something. Food or a clean litter box is pretty much the only things that she cares about. And then obviously at some point, you've got to stop it, right? So as we said, the elongation part is relatively mundane. Essentially, the initiation part is extraordinarily complex. And it's the termination. Termination is at kind of the other end. And termination really depends on eukaryote versus prokaryote. So prokaryotes have a couple of different ways of doing so, and eukaryotes have a related kind of but different way. So essentially, termination bacteria depends on those secondary RNA structures from forming. Right. Actually, just as a hold up for a little bit, anybody have any questions about what we've just covered in terms of initiation, promoters, stuff like that? Just to give you time to kind of percolate. No? Okay. All right. So there's two forms of termination. There's row-dependent termination, which uses a protein, and there's intrinsic termination. And so essentially these both these mechanisms rely on secondary structures in RNA and essentially levering the RNA polymerase holoenzyme off of the DNA, right? So if you can think about your RNA polymerase going kind of along there, we need something to go poink and like flick it off of the RNA. And the problem is that RNA polymerase is pretty fast, right? So it goes along, and anything behind it is like trying to catch up to shut it down. So both levering it off is one aspect, but also slowing down RNA polymerase so it can be levered off is the other one, right? And so we'll talk about these two particular approaches just now. Okay, and so part of that slowing down comes from, and this is very much the case in bacteria, it's much more complex in eukaryotes, and we'll get to eukaryotes next class on Tuesday, right? And again, this is like the general theme, right? It's relatively well defined, pardon me, in bacteria, and much more varied and so much harder to define in eukaryotes. And so in bacteria, quite often the thing that causes that to RNA polymerase slow down is a series of U's. Well, the series of A's in the DNA sequence, and then this poly U sequence in the RNA polymerase, right? And so that essentially causes that RNA polymerase to pause. It's almost like it's not really sure like where it should go, right? Have I finished? Do I keep doing? Have I done one already? Right? And so what then happens is we get what's called a terminator loop, right? Or a hairpin loop forming. So hairpin loops are inverted repeats, right? They're palindromes again. And so essentially at the very end of the transcript, or the gene, or the bit that needs to be transcribed, transcript, I guess, you have two features. This is intrinsic termination, right? So it doesn't require any extra factors. It's hardwired into the sequence of the gene. And so the coding region is presumably like it's way over to the left, right? We're now talking about the very bit at the end of the transcript. And so we have two features. We have a string of A's, which will result in a poly U sequence. And that's essentially what slows down the RNA polymerase long enough for it to be levered off of the transcript. And then we have this palindromic sequence in the mRNA, or in the gene sequence essentially, which when transcribed into mRNA kind of forms a hairpin loop, right? So essentially you've got kind of a palindrome in DNA is a little bit funkier because we're talking about two different strands, right? But essentially what you have in the RNA transcript are complementary sequences going in opposite directions, right? So where that's an A, that's going to be a U. I don't have any more fingers I can use, right? And then there's a G, and then there's a C, and then there's a T or U, and then there's an A, right? So that once they're produced, they're free essentially to base pair with each other and form a hairpin loop, right? Also called a terminator loop. And essentially what happens is that if you look back up here, right, this RNA polymerase holoenzyme is pretty big, right? And so what happens is that hairpin loop actually forms within the, within this channel of the RNA polymerase, right? So essentially the RNA polymerase pauses over one particular bit of DNA because of that poly U sequence. The mRNA that is produced forms that, I don't know how I can do it with my hands, right? That loop structure from base pairing of the RNA. And in doing so, it kind of takes up more space, right, inside that channel than is available. And so it kind of pops the RNA polymerase holoenzyme, right, because remember, it's a bunch of different proteins, apart, and then it dissociates from the DNA. The DNA kind of coils back again on itself, and all the different bits of the holoenzyme separate. Right, so again, pausing because of the U's, loop formation because of those inverted repeats. How's that work for you? We actually don't have too much left to cover today. We might be able to get started on the next one just to kind of free up a bit of space next class on Tuesday. Okay, so now we have row-dependent termination. So again, we have the same deal, right? We need a pause, right, to slow the RNA polymerase down to do whatever we're going to do to it. And we have a something, in this case, rho, which is a protein, which will disrupt the holoenzyme and the mRNA. And so the first part is, let's come back here. Here we go. Is we have a binding site for Rho, right? This protein. So again, that's encoded in the actual gene sequence of that bacterial gene, right? It recognizes that end essentially of, or near the end of that transcript. And what we mean by unstructured RNA is that it doesn't have any of these kind of funky 2- and 3-dimensional structures like this hairpin loop, right? So again, and this terminator sequence here is again a loop, right? Doesn't actually force the holoenzyme apart in this case, but what it does is it slows down RNA polymerase, right? So it pauses. And basically that allows Rho to catch up and leave a RNA polymerase off of the DNA strand. And it does so basically by, remember we talked about how the mRNA transcript is bound like by base pairing for a short stretch, and then it kind of dangles off in the breeze. Rho will actually force the hybrid apart, and as soon as it does, then transcription cannot continue. So again, pausing via this terminator sequence, which forms a loop, we'll come across that again when we look at trip operon regulation, right, which we'll start covering next Thursday, maybe. So we've got a pause with the terminator sequence, which is a loop in this case. And then we have a second thing, which is our Rho, which is traveling along the mRNA as fast as the RNA polymerase. So it never catches up essentially until that RNA polymerase is slowed down by that terminator sequence. And then Rho is able to catch up and do its job. Any questions on that stuff? This is going through a little fast. The trouble is there's quite a lot of information here. So I don't want to kind of race on too much. But also it's, I don't know, pacing is difficult with stuff like this. All right, if you do have questions, again, just shout them out or write them up. It's all good. Okay, so in this is in eukaryotes. Yeah, this is eukaryotes. We have again the same kind of idea, right? So RNA polymerase 2 in eukaryotes doesn't have a kind of a specific sequence, right, which pauses it. Instead, what we have is a it's kind of like basically degradation based. So in bacteria, we have a slowdown of RNA polymerase and something which forces RNA polymerase off of the DNA, or the RNA off the DNA, which is essentially going to be the same thing. In eukaryotes, what we have is there's no particular sequence, right, which kind of slows down RNA polymerase or causes it to pause or anything. Instead, what we have, this is part of the RNA processing apparatus, right? Because in prokaryotes, it's quite simple. You get an mRNA, it gets translated, right, pretty much immediately. In eukaryotes, we have a whole bunch of different steps to process that transcript into its final functional form, actual mRNA, right? And before that happens, it's called a pre-mRNA, which you can see up here. And so essentially there's going to be a cleavage site, which is going to be a very specific sequence in the transcript, which is recognized by an enzyme, which will come in and it will cleave inside of that, right? So this is an endonuclease. Well, that's, and it calls itself an endonuclease. Oh, I don't know, whatever. don't know, that would be, I'll call that an exonuclease. But anyway, it's an enzyme which recognizes and can cut nucleic acid kind of in the middle of a stretch of sequence. So that is important for two reasons. One, it frees off the mRNA, and this will then have a poly-A tail added to it, which is like one of the parts of the processing. And so that pre-mRNA, depending on how much is left to do with it, is now done, right? The RNA polymerase doesn't know this. Again, they're kind of dumb as a box of bricks. So it'll just keep on transcribing pretty much forever, right, if it's allowed to. However, now, and we'll see why this is important later in eukaryotes, now we have a free end, right? This is now able to be attacked by another enzyme, which in this case is called RAT1. Don't know why they call it an endonuclease. I'd call it an exonuclease personally, because it attacks the end of a strand, right? Typically, you don't find those in mature mRNAs, right? Because they're protected at either end. And so that free end is basically ripe for being chewed up. And so RAT1 binds to it, basically finds any free 5' end of mRNA and will attach to it, or RNA in general. And it will start degrading that mRNA sequence, or that RNA sequence rather, sorry, because it's not mRNA anymore. It will start degrading that RNA sequence until it catches up with RNA polymerase, right? And so essentially it just goes, chops it all up and kind of forces its way in as it's trying to chew up that very last little bit of RNA that the RNA polymerase is making. It kind of gets into that channel and it forces the RNA polymerase off of the DNA, essentially. And so that's how transcription termination occurs in eukaryotes. There isn't a specific end sequence as such as there is in prokaryotes, or if there is, we don't know what it is yet. Instead, there's all right, this is the end of the functional transcript. Everything after this point is junk, it's not needed. And that cut point, that's what serves as the basis for the termination by RAT1, right? Doing it by degradation, not by moving along the RNA as Rho does in bacteria, but by actually just chewing it up. And presumably that's why it's faster than RNA polymerase, and it doesn't need that slowdown. Okay, anybody got any questions? Okay, so what I want to do now is change gears and look at RNA processing in eukaryotes. Let's see if we can make a little bit of headway on this. Because if we do, then that might free up a little bit of time on Tuesday for recap, if you have any questions. So if you are able to, try and study the material that we've covered between now and, pardon me, Tuesday. Also have a look at the PowerPoint we're just about to start. Let's see, which is, come on, this one? And then bring those questions with you for Tuesday, and hopefully we'll have a little bit of spare time to go over stuff before the exam on Thursday. Right, so again, last stretch, is not going to be much more, just got to hold it together until the end. So essentially, there's two, well, always get careful making numbered lists. There's a few kind of really important things that need to happen in eukaryotes in terms of mRNAs. So mRNA, as I said, is very vulnerable to degradation, right? It doesn't hang around very long. There are loads of RNases around. Actually, there's loads on your skin, believe it or not, which I think they protect against RNA viruses. Not sure, but there's loads all over you. But RNA is very fragile, right? And so one of the ways it is fragile is it's open to attack from either end, right? So if you think back to that RAT1 nuclease, right? Any free 5' end in the cell, it will attach itself to, and it will just chop up that mRNA, right? So one of the big parts of RNA processing in eukaryotes is to protect that mRNA. And so that's what we're going to use. Basically, the 5' and the 3' end, we're going to stick stuff on those ends so that they're no longer recognized by those nucleases and that won't be degraded, right? So that's kind of one big deal, right? Is protecting those mRNAs from degradation before they're supposed to be degraded, right? Because they will be degraded at some point. The second big deal, the second big deal is to get rid of the introns, right? So if you remember back to, let's see, this one. Where are we at? Here we go. Right, so if we were to make a transcript from this and then translate it, we would have a whole bunch of stuff in there which shouldn't be made into protein, right? And that'll essentially, it'll cause an awful lot of problems. It'll either produce a gibberish protein, or there will be a stop codon hiding in there which will just terminate translation early, right? So you won't get your functional protein. Now, we need to remove those introns to make a finished final mRNA, mature mRNA transcript that can be exported to the cytoplasm and serve as a template for translation, right? There are other things going on. So essentially, as part of that process of adding our cap to this end and our poly A tail to this end, those ends actually have a role in both translation and mRNA stability, which we'll see later, not today, but we'll see that on Tuesday. So there's a lot of stuff going on here, right? So we need to protect the ends. We need to get rid of that crap in the middle, which we don't need, right? And we need to do it very precisely too. And those bits at the end will also be really important for kind of initiation and control of translation and other stuff as well, right? So as you can see, there's quite a bit more going on with eukaryotes than there are prokaryotes. Oh, I'm sorry, like end of the week type stuff, I guess. So literally as soon as the 5' end of the transcript emerges from the RNA polymerase holoenzyme, it gets a cap put on it, and that's called a 7-methylguanosine cap, or 7-MPG. Right, and so essentially it's a it's kind of a nucleotide, sort of, but it's modified in a way which makes it unrecognizable to enzymes. And also, oh, actually, here we go. We've got found a use for that 2' OH. Forgot about this. You also have other methyl groups added to other bases, right? So it's essentially, it's a clump of unrecognizableness, right? And this whole bit is added to that 5' end as soon as it emerges, right? So you literally have your holoenzyme here, dang, 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 chungling along. And then you have the capping enzymes are hanging around. So as soon as the mRNA comes out, comes in, sticks the cap on, and now that end is protected from exonucleases that happen to be floating around, right? It's not going to get degraded, either during the process of transcription or later on once it's been exported, right? So this 7-mpg cap is really, really important. It's also involved in assembly of the ribosome and translation as well. Now we have a couple of other things that need doing. I'm going to do splicing first, and then we'll come back to the poly A tail, because it kind of makes more sense that way around. Okay, so you hold your horses. I'm going to do some of this. We'll see how much we get through. So essentially what we need to do now is, let's draw this up actually. This is going to be a little bit easier to draw than it will be to describe in words. A red pen go. Oh, maybe I'm sitting on it. All right. Black and green are it. So I can, perhaps this is where I really need my large oak desk. So I'm going to draw this in blocks. It doesn't actually exist in blocks, obviously. This is more of a kind of conceptualization. And so we have our cap here, our 5' MPG. We have our 5' UTR, right? So remember, that's our untranslated region. This is also important for ribosome assembly. Kind of just gives it a bit of space, basically. Then we have, and this is going to be AUG. Oops, crap. Writing real small with these things is not trivial. Right, so that's our start codon. And we'll get back to what codons are and the like. And that's our first exon. Right, so as this is, I don't know, I think it might be that there's background noise in Abigail's environment, which keeps popping up like it's she's speaking. Sorry, I can't do much about that, unfortunately. It doesn't last for very long, though. Okay, so I'll just keep on talking. Drown her out, right? So essentially what's being produced is, and then we have our RNA pol over here, right? So this is, don't worry about it, Abigail. Everybody has a different environment that they're working in. So essentially this is going on while transcription is actually occurring, right? So obviously the very first thing that happens is that that cap gets added. And then the second thing is, as soon as these introns start appearing, and it doesn't actually have to be in a linear fashion, right? It could be the intron, so this is intron 1, this is exon 2, right? It could be the intron 2 is the first one that's taken out. Doesn't really matter. But as soon as these start appearing, now they're going to be available to be spliced out, right? And so this is very much the same as what one would do with old cinema film. You know, if you want to cut out a scene, you would cut it, remove the bits you want with scissors, and then you just stick it back together again. Or if you've watched Fight Club, then you could do other things in films. But anyway, so little bit of a, you know, a cultural reference there, right? So what's going to happen is that we're going to recognize the beginning and the end of these introns hang on, what? Oh, I can't remember. I've forgotten. Pretty sure it's AU. I'm gonna have to check now. I used to know this stuff right off the top of my head. It's in here. There we go. GUAG. That's it. There we go. Sorry about that. Sometimes feels like I have so much stuff stuck in my head that you know, things just get jumbled around and lost. But essentially what we need to do is we need to recognize the start of that intron and the end of that intron. Because what we want to end up with is exon 1 and exon 2 stuck together seamlessly, right? No extra bases in there, no bases removed, because otherwise the whole reading frame of that those two exons will be screwed, right? And you'll get the wrong protein as a result. So first up, these are going to be recognized, and we'll talk about some of the details in a little bit. And then what you want to end up happen is that intron, and then RNA polymerase is still over here somewhere. Right? Now we have exon 1 and exon 2 together, and intron 1 has been cut out, right? It's just be degraded basically and recycled, and those nucleotides will be reused, right? So there's a bunch of things that need to happen for this to work. One, we've got to recognize these, they're called splice acceptor sequences, and they're really, really important, because if you have mutations in these, then that intron won't get spliced out, and you will have a non-functional protein as a result. There's also, I think it's an A, another nucleotide in here. These are basically consensus sequences, so that's what, there's other stuff in there as well, which helps. We're then going to bend this transcript around using that A. I'm pretty sure it's an A. Let me just double check. Yeah, there we go. We're going to bend it around, and we're going to essentially cut our mRNA or pre-mRNA, right? Right, because this is pre-mRNA until it's all finished, right? It's only once the mature mRNA has everything done to it, you know, capped, all exons removed, poly-A tail on the 3' end, is it considered an actual mRNA. Until that point, it's still not usable, right? And it won't be exported from the nucleus until all of that stuff has been done, right? So we need to recognize that. We're going to bend it round, and we're going to make a cut just here and just here, and stick those bits of mRNA back together again, right? That's how we do our splicing. And this is called splicing. Ta-da! Not splinching. That's from Harry Potter. Similar principle though, I guess. All right, let's take a picture of that before I forget. Any questions while I'm doing this? Again, it pays to keep in mind the bigger picture while you study this, because otherwise the little details can get overwhelming. Okay, let's just make another 10 minutes or so I don't blow your brains out your ears, and give us time to have lunch before lab starts at 2. Okay. No, sorry, I try. Okay, so again, this is remember the term pre-mRNA is an important one, right? Because it's basically telling you it hasn't been finished yet. And so what we're going to do is we're going to have a bunch of proteins come in. These are going to be called SNRPs. This is a really annoying word to write out, so I'm only going to do it once. SNRPs. Basically small nuclear ribonuclear nucleo. Sorry. That's why I call them SNRPs. It's a lot easier. So basically what that long ungainly word tells you is that the ribonucleoprotein means that it's a mix of RNA and protein, right? Hence ribonucleo and protein. They're small because they're small, and they're nuclear because they're found in the nucleus. That's why they're small nuclear ribonucleoproteins. There. Try saying that fast after you've had a few. Anyway, so these SNRPs, and there's a whole bunch of them, which you'll see down here, U1 to 6, I think. Their job is to come in and do all of those functions, right? It is to recognize those splice sites. It's to cut the intron or exon 1, intron 1 boundary. And it sticks that intron back to itself, right? So again, if you kind of tilt your head and look at the insert on the right, okay, so the bit along the bottom is the, you know, the linear bit of intron. The 5' end is looped around, and then it actually forms a phosphodiester bond with the 2' OH of that one of the nucleotides in that strand, right? So that forms what's called a lariat loop, perhaps because it looks like, you know, Texans chasing after cattle with a lasso or something, right? And this is what it looks like in painful detail, right? So essentially you have U1 and U2, right? The U1 is involved in finding that 5' splice acceptor. U2 is the one involved in finding the branch point, right? And they're involved in bending round the RNA structure, and kind of there's a whole bunch of other SNRPs in there which form a complex, right? This complex is called the spliceosome. Let's write that out because it's fun. Spliceosome. All right. And essentially what it needs to do, and this is a fairly complex thing if you think about it, it needs to basically cut exon 1 off of the mRNA, but it can't lose it, right? Because if that exon 1 floats off into the rest of the nucleus, you're screwed, you lost it, right? It's like, oh crap, it's gone, right? So not only do you need to cut off exon 1, you also need to hold it there, right? So it doesn't disappear, right? And then at the same time, you need to cut the boundary of the intron 1 and exon 2 and stick exon 1 and exon 2 back together again, right? Once that's done, the intron and the SNRPs wander off to be reused, degraded, whatever is their fate, right? To repeat the same thing again. So every intron that is present in this pre-mRNA has to have this occur to it, right? So that you can get rid of them, right? And there can be multiple exons, varying sizes, right? In some genes, you can have, you know, 10 or 12 introns, and they're huge, right? So the exons are like these little bits dotted along the transcript, and these introns, these huge spaces in between. Anybody got any questions about that? There's quite a lot of stuff to take in, right? But again, it pays to remember the general principles. We've got to find the intron. We've got to cut the intron and the exon, and then we've got to stick back those two introns together so that it makes one continuous mRNA sequence. You all good? All right, and then just to finish up, let's go back up here and look at the last thing that has to happen. We can recap this on Tuesday too, just to kind of go back over it, make sure we haven't left anything out. And so the last thing to do, remember I told you that the 5' cap is really important. The same thing applies to the 3' end, right? And so you don't want to leave that bare because then it will be degraded by nucleases or RNases in the cell. And so essentially, and this again is just a eukaryotic thing, doesn't happen in prokaryotes, right? We need to add a short sequence of A's. This is our poly A tail, right? And so if you remember in the termination of transcription, we have a cleavage site, right? So essentially we have a spot that is going to be cut to separate the pre-mRNA from the I don't know, leftover tail, which is going to be degraded by RAT1. So as soon as that spot is cut, and we know which site to cut based on the sequence of consensus sequence, right, which the enzyme recognizes. As soon as it cut, another enzyme comes in and adds a long string. Could be, I don't know, how much are they long? Between 20 and 100 base pairs, usually like 30 to 50, give or take, of just A's, nothing else, right? Called polyadenylation. It does not require template, right? So this isn't a copy of any sequence that's in the DNA. It's simply a repeat. There's an enzyme just comes in and goes A, there's nothing else, right? It's called polyadenylase, funnily enough. All it does is just adds a string of A's until it gets bored of its job and it falls off, right? But that string of A's, that poly A tail, serves as a binding site for what are called poly A binding proteins. And those are what protect the end, the 3' end of the pre-mRNA transcript. Let's just write that down so we have that commemorated. Right, so it's not actually the poly A tail itself which protects the 3' end. It's the proteins that bind to it. So that adding the tail is essentially a way of providing a place for those proteins to bind, right? And modifying how well those proteins are bound or not is actually one way we can control stability of the transcript. It's, you know, another extra wrinkle, right? And I think that's more than enough for today. We've actually made some really good progress. So that should hopefully free up some time on Tuesday where we can finish translation. We'll recap RNA processing too, because it's quite a lot to take in one go. Do translation, which is big, and then have time to recap any of the material for the exam on Thursday. All right, cool. So let's meet back at 2 p.m. for lab. See you all in a little bit. Enjoy your lunch. I'm going to see what I can rummage around and find in the fridge.